Good morning, everyone. Today, we'll be exploring two core immunological concepts, affinity and avidity. These terms both describe how antibodies bind to antigens, but they capture very different aspects of that binding. Affinity refers to the strength of a single interaction. Imagine one lock and one key fitting together. Avidity, on the other hand, reflects the combined strength when multiple locks and keys are engaged simultaneously, as happens in real biological systems where antibodies and antigens are often multivalent. Why does this matter? Because the effectiveness of our immune response hinges not only on whether antibodies recognize antigens, but on how strongly and stably they bind. High-affinity antibodies are essential for neutralizing pathogens effectively. Avidity explains how antibodies like IgM, which individually have modest affinity, can still be powerful defenders thanks to their pentameric structure that allows multiple simultaneous interactions. In this lecture, we'll unpack the molecular basis of antigen, antibody binding, define affinity and avidity in detail, contrast them side by side, and then look at their biological and clinical importance. By the end, you should not only know the definitions but also appreciate their implications. Why vaccines rely on affinity maturation, why avidity testing can tell us whether an infection is recent or old, and why both concepts are indispensable for interpreting laboratory assays like ELISA and agglutination tests. So, let's begin by reviewing the fundamental principles of antigen-antibody interactions. Before we dive into the heart of today's lecture, let's pause and set out our learning objectives. First, you'll be able to define affinity and avidity with precision. This may sound basic, but in immunology, definitions are powerful tools. Affinity and avidity are often confused, even in research discussions, so being crystal clear on what each term means is the foundation for everything that follows. Second, you should understand the molecular basis of antigen-antibody binding. These interactions aren't based on covalent bonds. They rely on weaker, non-covalent forces such as hydrogen bonding, electrostatic attractions, van der Waals forces, and hydrophobic interactions. Alone, each of these forces is weak. Together, they can create very stable and highly specific interactions, like the cumulative effect of Velcro hooks holding onto loops. Third, we want to differentiate between affinity and avidity with practical examples. Knowing definitions is only the first step. Being able to apply them is what demonstrates real understanding. For example, we'll compare IgG, which has high affinity but only 2 binding sites, with IgM, which often has lower affinity per binding site but achieves very high avidity thanks to its pentameric structure. These examples anchor abstract concepts in real immunological situations. Fourth, we'll explore the biological significance of these binding strengths. This is where molecular detail meets physiology. You'll see how affinity maturation improves the immune system's precision over time, and how avidity explains the effectiveness of early IgM responses before affinity has been optimized. This connection to immune defense and vaccine responses shows why these terms are more than definitions, they're functional realities. Finally, we'll apply these concepts to laboratory tests like ELISA, agglutination, and avidity assays used in infection diagnosis. Antibodies, or immunoglobulins, are Y-shaped proteins produced by B cells. At the tips of the Y are the antigen-binding regions, called Fab regions. Each Fab contains a unique variable domain, which determines the antibody's specificity, its ability to recognize one particular molecular structure, or epitope, on an antigen. It's important to note that antibodies don't form covalent, permanent bonds with antigens. Instead, their interactions are based on non-covalent forces, hydrogen bonds, ionic interactions, van der Waals forces, and hydrophobic effects. These are individually weak, but when several occur in just the right alignment, they create a strong, specific, yet reversible interaction. This reversibility is crucial. It means antibodies can bind tightly when needed, but they can also release, which allows the immune system to adapt and refine its responses. The strength of this interaction depends on both structural complementarity, the fit between antibody and antigen, and chemical complementarity, meaning the compatibility of charge and polarity. Another important point, antibodies are bivalent at minimum. meaning each molecule has two identical antigen binding sites. Some classes, like IgM, are even more multivalent, existing as pentamers with 10 potential binding sites. This multivalency is what allows us to distinguish between affinity, the strength of one site binding one epitope, and avidity, which considers the collective binding when multiple sites are engaged simultaneously. So, to summarize, antibody-antigen binding is specific, reversible, and governed by multiple weak forces acting in concert. It is this delicate balance of specificity and reversibility that underpins the entire concept of immune recognition, and it sets the stage for understanding how affinity and avidity emerge as measurable properties of these interactions. Now that we understand the basics of how antibodies and antigens interact, let's focus on the first key concept: affinity. In immunology, affinity refers to the strength of binding between a single antigen binding site on an antibody and a single epitope on an antigen. You can think of it as the molecular equivalent of a handshake. How firmly does one hand grip the other? If the fit is tight and stable, we describe the interaction as high affinity. If the fit is loose and easily disrupted, we call it low affinity. Affinity is an intrinsic property, meaning it reflects the quality of one binding site interacting with one epitope, independent of any other sites. It's governed by the same non-covalent forces we discussed earlier: hydrogen bonds, van der Waals interactions, hydrophobic contacts, and electrostatic attractions. The overall balance of these forces determines whether the antibody and antigen will form a stable complex or whether they will quickly dissociate. From a quantitative perspective, affinity is expressed using equilibrium constants. The association constant, Ca, reflects how easily an antibody and antigen come together, while the dissociation constant, Kd, reflects how easily they separate. A low Kd corresponds to high affinity because it means the complex is stable and dissociation is unlikely. These values can be measured experimentally, for example, using surface plasmon resonance or equilibrium dialysis, which allow us to assess the binding strength of individual antibody-antigen interactions. Biologically, affinity matters because it determines how effective an antibody will be at recognizing and binding its target even at very low antigen concentrations. During the course of an immune response, B cells undergo a process known as affinity maturation, in which mutations in the antibody's variable regions are selected for stronger binding. This process ensures that, over time, the immune system produces antibodies with progressively higher affinity, which are far more effective at neutralizing pathogens. So, to put it simply, affinity is about quality, the strength of one-on-one binding. It is the foundation upon which the broader concept of avidity is built, and it plays a critical role in shaping how precise and powerful an antibody response can be. Having established what affinity means, let's now shift to the broader concept of avidity. Whereas affinity describes the strength of a single antigen binding site interacting with one epitope, avidity captures the overall strength of all interactions between a multivalent antibody and a multivalent antigen. In other words, avidity reflects the functional reality of antibody binding in the immune system, where multiple sites are almost always engaged at once. A good analogy here is Velcro. One hook and loop connection represents affinity, weak and easily separated. But when you have dozens of hooks and loops engaged together, the combined effect is strong and very difficult to pull apart. That is avidity. Even if each individual interaction isn't especially strong, the cumulative effect of many simultaneous bonds creates remarkable stability. Antibody structure plays a central role in determining avidity. IgG molecules, for example, have two antigen binding sites, so they can engage two epitopes simultaneously. In contrast, IgM, which circulates as a pentamer, has 10 potential binding sites. Even if each site has only modest affinity, the overall avidity of an IgM molecule is extremely high because of the sheer number of simultaneous interactions. This is why IgM is so effective in the early stages of infection, when antibodies haven't yet undergone affinity maturation. Avidity is not only about the number of binding sites, though. It also depends on the valency of the antigen, how many epitopes are available for engagement. A multivalent antigen, like a bacterial surface with repeated epitopes, allows for high-avidity interactions. By contrast, if an antigen is monovalent, even a pentameric antibody cannot achieve strong avidity because there are not enough epitopes to engage. In practical terms, avidity is often a better reflection of biological reality than affinity. While affinity can be precisely measured for a single interaction, avidity describes how antibodies perform under physiological conditions. This is why avidity is particularly important in diagnostic immunology. For instance, avidity testing can distinguish between recent and past infections. Early antibodies tend to have low avidity, while antibodies produced later, after affinity maturation, have high avidity. To summarize, avidity is about collective strength. It integrates affinity with antibody structure and antigen valency to give us a more complete picture of antigen-antibody interactions as they occur in the real world. At this point, we've defined affinity and avidity separately, but the real value comes from comparing them directly. Affinity is the strength of a single interaction, one antigen binding site on an antibody and one epitope on an antigen. Avidity, by contrast, is the cumulative strength when multiple interactions occur at once. This distinction may seem subtle at first, but it has profound consequences for how antibodies function in vivo and how we interpret their effectiveness in the lab. Think of affinity as quality and avidity as quantity plus cooperation. A single IgG molecule may have very high affinity for its specific antigen, meaning one Fab arm grips an epitope tightly. But IgM, even if each individual binding site has only moderate affinity, achieves tremendous avidity because it can bring 10 sites to bear on the same or repeated epitopes. The result is a binding strength that is far greater in practice than you would predict by looking at a single interaction. Another way to compare them is by reversibility. A high-affinity antibody may hold onto its antigen tightly, but if it has only one or two binding sites engaged, the complex can still dissociate under certain conditions. With high-avidity binding, however, dissociation is far less likely because multiple interactions must be broken simultaneously. This creates a kind of biological glue that stabilizes antigen-antibody complexes in physiological environments. We also see the difference in how they're measured. Affinity can be quantified directly using equilibrium constants such as Ca or Kd, providing precise numerical values for the strength of one interaction. Avidity, however, is more difficult to express in strict mathematical terms because it depends on both the affinity of individual sites and the valency of the antibody and antigen. Instead, avidity is often assessed functionally, for example, by observing how well antibodies resist dissociation in laboratory assays. From a biological perspective, both are critical. High-affinity antibodies are the product of affinity maturation, giving the immune system precision and efficiency. High avidity, on the other hand, allows early immune responses, particularly those dominated by IgM, to be effective even before affinity maturation occurs. So, when comparing affinity and avidity, remember, affinity is the strength of one hand gripping, while avidity is the combined grip of many hands working together. Both concepts complement each other, and both are essential to a full understanding of antibody function. Now that we've compared affinity and avidity conceptually, let's ask a deeper question. Why do these properties actually matter to the immune system? Understanding their biological significance allows us to connect abstract molecular ideas with the real-world function of immunity. Let's begin with affinity maturation. When B cells are first activated, the antibodies they produce typically have moderate affinity for the antigen. As the immune response progresses, these B cells undergo a process in germinal centers called somatic hypermutation. Mutations in the genes encoding the antibody's variable regions slightly alter the structure of the antigen binding site. Only B cells that acquire mutations improving the fit between antibody and antigen are selected to survive and proliferate. Over time, this evolutionary process leads to antibodies with progressively higher affinity. The result is a stronger, more precise immune response capable of neutralizing pathogens effectively, even when antigen concentrations are low. Now consider avidity. Its importance is especially evident early in infection. The first antibody isotype secreted is IgM, which forms a pentamer with 10 potential binding sites. Individually, each binding site may not grip very tightly, the affinity is modest. But together, the multivalency creates exceptionally high avidity, making IgM highly effective at agglutination and complement activation during the initial stages of infection. Thus, avidity ensures that the immune system is not defenseless while affinity maturation is still taking place. From a functional perspective, high-affinity antibodies excel at neutralization, blocking viral entry into cells, inhibiting toxin activity, and tagging pathogens for destruction. High avidity, on the other hand, provides robustness, ensuring that complexes remain stable even under physiological conditions where dissociation might otherwise occur. This stability is critical in clearing large pathogens or forming immune complexes that can be processed efficiently. Clinically, both properties have implications. Vaccines, for instance, aim to stimulate affinity maturation, leading to long-lasting high-affinity IgG antibodies. Meanwhile, the concept of avidity explains diagnostic patterns. Newly produced antibodies often have low avidity, while mature responses display high avidity, which can be used to distinguish acute from past infections. In short, affinity fine-tunes the quality of immune recognition, while avidity ensures strength and stability of binding in real biological contexts. Together, they illustrate how the immune system balances precision with power. Having explored the biological significance of affinity and avidity, let's now turn to how these concepts are applied in laboratory and clinical settings. One widely used method is ELISA, the enzyme-linked immunosorbent assay. In an ELISA, the intensity of the signal depends on how strongly antibodies bind to their antigens. High-affinity antibodies bind more stably, producing stronger signals at lower concentrations. Similarly, avidity affects the outcome. Antibodies with multiple binding sites that engage multivalent antigens produce more robust, stable complexes, which can improve assay sensitivity and reliability. This is particularly important in detecting low-abundance antigens or distinguishing closely related antigens. Another classic example is agglutination assays, such as those used in blood typing or detecting bacterial infections. Here, the visible clumping of particles occurs when multivalent antibodies cross-link antigens. High-avidity antibodies excel in these assays because multiple simultaneous interactions create large, stable aggregates that are easy to detect visually. By contrast, antibodies with low avidity may bind individually but fail to form observable clumps. Affinity and avidity are also crucial in infection timing diagnostics, such as for torch infections or viral diseases like cytomegalovirus. Early in infection, antibodies tend to have low avidity because they have not yet undergone affinity maturation. Over weeks to months, avidity increases as the immune system refines antibody specificity. Finally, these concepts inform vaccine evaluation. Successful vaccines aim to elicit high-affinity, high-avidity antibodies that are both specific and robust, ensuring long-term protection. Measuring the quality and functional strength of antibodies after vaccination can help predict efficacy and guide booster strategies. To wrap up today's lecture, let's review the key points and reinforce the concepts of affinity and avidity and why they are central to immunology. First, affinity refers to the strength of a single antigen binding site interacting with a single epitope. It is an intrinsic property of that specific interaction and is determined by structural and chemical complementarity, as well as the non-covalent forces holding the antibody and antigen together. High-affinity antibodies bind tightly and stably, while low-affinity antibodies bind weakly and are more easily displaced. Affinity is critical for the precision of the immune response, and it improves over time through the process of affinity maturation, which occurs in germinal centers during adaptive immune responses. Second, avidity represents the overall functional strength of binding when multiple antibody binding sites engage multiple epitopes on the same or multivalent antigens. It is a product of both individual site affinities and the valency of the antibody and antigen. Avidity explains why IgM antibodies, despite having relatively low affinity per binding site, can be highly effective early in infection. It also provides stability to antigen, antibody complexes in physiological conditions, making it biologically significant. The comparison of affinity and avidity highlights that while affinity describes the quality of a single interaction, avidity reflects the combined strength of multiple interactions. Both are essential. Affinity ensures specificity and effective pathogen neutralization, while avidity ensures stability and robustness of binding. Finally, these concepts are directly relevant in laboratory and clinical applications. High affinity and high avidity antibodies improve ELISA sensitivity, support agglutination assays, inform infection timing and avidity tests, and guide vaccine evaluation. Understanding these principles allows immunologists and clinicians to interpret antibody function and the immune response accurately. In conclusion, affinity and avidity together provide a comprehensive picture of antibody effectiveness. Affinity fine-tunes the precision of the immune system, avidity strengthens its functional power, and together they form the foundation for both our understanding of immune defense and practical applications in diagnostics and therapeutics. Keeping these distinctions in mind will help you connect molecular immunology to real-world biological and clinical contexts. Thank you for watching. Subscribe for more.